Thank you, Maggie. Can everyone hear me? Okay, awesome. Well, thank you for the introduction, Maggie. I'm Katie Childers. Like she said, I'm starting my 6th year as part of the bioengineering program here at KU. I have the very unique pleasure to talk to you about integrated multi-scale fluidic systems. I would just like to specifically mention that this is adapted from Professor Dr. Murphy, who passed this year. And so I just want to bring attention that he did a lot of the work towards these integrated systems for our center. So for this presentation today, I'm going to introduce integrated systems, what they are, what they look like. I'd like to introduce some of the peripherals, specifically looking at it from a systems design consideration perspective, then give a kind of a survey of topics in terms of what is out there in the literature, and then what we actually do in the center for building our microfluidic platforms. And then I'll finish with actually a case study of an integrated system that we're currently working on in the lab now. So what are integrated mixed scale fluidic systems, or what is their purpose? Generally, for assays to go from sample in to answer out, they increase in complexity. And so you need to be able to basically have more complex assays contained in a small space. And so integrated systems kind of have two purposes. One is accelerating research. You can think of lab on a chip, the idea of doing the full assay all on one single kind of system or chip, as well as personalized medicine. And really the focus of my talk is going to be more on in vitro diagnostics and point of care testing, but this can extend out to many different applications, which I'll summarize later. But this really started with the idea of a micro total analysis system, basically housing all of the peripherals for an assay all on a single system. There's 3 different main types of microfluidic systems that we like to classify. It's really based on the fabrication method and then how you're moving fluid throughout the system. There's centripetal, monolithic, and modular. So centripetal is basically this lab on a disc technique. You're housing all of your fluidic system on basically a CD or DVD, and using centripetal forces, basically spinning on that middle axis, rotating that disc, using centripetal forces to move fluid throughout that system. Now that limits the amount of space you can actually use or have on this disc, which increases the fabrication complexity and limits what you can do on that. Monolithic, the idea is it's all on a single substrate. It's a monolithic substrate where you're housing all of your fluidics. So the example I have here basically is a 2-step process. You're depleting blood and then isolating out CTCs that Maggie introduced in the previous talk. But this is all on one substrate. So when they fabricated it, they made all of the features on this one single chip that you see here. Now, if you want to redesign that, that increases time and effort that you would have to do to redesign and change the fabrication method. Now that limits the amount of applications that this one device can be used for and increases the cost of development. Finally, there's modular, and the idea is you have these different modules, kind of plug and play style, where you have these modules that basically plug into a fluidic motherboard, and each of those modules have a single step in the more complex assay. Now, this is more easily reconfigurable. You can mix and match the different materials and what fabrication method you're using, and the modules themselves can be used as standalone units. And I'll go more into modular and monolithic systems here shortly. Monolithic integrated systems is really kind of where the field started. I have some references from 2004 and 2005 on these monolithic integrated systems, and you can see the complexity of these. These integrated both fluidic systems with MEMS technology, so micro electrical mechanical systems. So on the one on the top, you can see this has pumps, valves, sensors, a PCR mixer. It's got everything housed in this one substrate, basically with the electronics sitting on the top. The one on the bottom here is very similar. It has all the fluidic connections as well as kind of all the electrical and mechanical attached to the top here. While these are sample in and answer out operations, they're really single targets. They're optimized for mainly one application. And as your assay increases in complexity, you're going to have to develop kind of more complex manufacturing processes to get this all onto one substrate in this monolithic setup. Although people are doing large-scale integration of monolithic systems, this is a pretty cool system. This is using over 2,000 valves that are housed in this single chip, and it has basically 250 picoliter-sized little reaction chambers on here, where all this color is just food dye, just to show you the complexity. But they were looking at the expression of an enzyme and controlling all these valves pneumatically. And so this is an example. So people are looking at large scale buildup of these monolithic systems, but again, you're limited based on your application and your fabrication method. So that's where I want to draw the difference between kind of monolithic and where we're headed more in a modular approach. So monolithic, again, you're containing it all on a single chip, whereas modular, you're basically having functional units kind of separate, optimized separate, and then brought all together onto a single platform that you can kind of see here. This is 4 different chips kind of surrounding this separate chip. So you can do different applications on each of these chips and then fluidically direct them to the final readout sensor in the middle here. Again, the advantages of modular is that it has a lot of versatility. You can kind of think of it like Lego blocks for biologists or chemists. They can kind of plug and play and build their assay in different ways. It's very scalable in that you can use manufacturing practices for each of the functional units that would be best for a large scale production. And you can optimize each process separately in each of these functional units. So modular systems have been growing in research and development and academia and patents as well. You can see this is the number of modular microfluidic papers and patents over the last 12 years. So there's 2 different kind of configurations I'd like to introduce for modular systems. There's one's like a motherboard. If you're familiar with like electronic systems, it's kind of like plugging in different electrical components onto an electrical breadboard or circuit board. So each of those functional units has a function and it's being plugged in and connecting electrically. But for microfluidics, obviously, it's being connected fluidically. And this could look something like this, where you have a motherboard and you're basically plugging in your different functional units. Or you can have stack puzzle, and I'd like to add another one here. It's kind of Lego block-like. There's lots of work being done on actually using Lego-like connections to connect these different modules together. But you can you have the flow between the modules. There's not necessarily the central motherboard, but you have the different functional units that are able to connect together, flow together, and create more complex assays. And I've included some references below that are examples of these motherboard versus stack puzzles. So how do we at CBM² actually think about these integrated and modular systems approaches? Maggie had a great talk on these different isolation techniques for CTC and nanoparticle isolations. So these do basically just the isolation step, like she said, the sample prep step. Now, something like the in-plane nanopore sensor, which you've heard a lot about and are going to be making and running, this is more of a detection method, right? So you have your resistive pulse sensing, you're you are detecting the differences in these different analytes by flowing them through the nanopores and basically seeing the difference between the analytes based on those characteristics. Now, those each do one step. But if we're thinking more in systems integration, you're building systems. So you're putting these different modules in series with each other to build up assays. 2 I want to draw attention to from the center are the smart chip and the genosensor. The smart chip used the CTC isolation and did immunophenotyping analysis downstream, all on this integrated motherboard. The genosensor was looking at drug-resistant tuberculosis using DNA microarrays. Again, you're putting these modules in series. And what's really great, again, just driving home, you can use a variety of fabrication techniques and materials for each of those modules. You can easily reconfigure, and I'll get into that kind of with the smart chip a little bit. And you can use the modules as standalone units. Again, you're going to be using the in-plane nanopore sensor for some testing this afternoon. And you can use the CTC isolation, like Maggie showed, separately from these systems. But when we're thinking about systems, we really have to think about the integration steps. So anyone familiar with kind of molecular biology or any analytical chemistry, you know all the different heating or electrical components that you need to actually have to run these assays. So the idea of systems is you have these different requirements like valving, mixing, voltage, heating, pumping, all allowing you to fluidically connect the different modules and actually run the assay and do the molecular biology on the different chemical assays. So with that in mind, I'd like to kind of transition to design considerations. Again, I'm mainly focusing on in vitro diagnostics or point of care testing, but these design considerations can adapt to whatever kind of application you're going for. But this is a really great review paper from 2017. You have to think about where your system is actually going to be deployed, right? So this broke it up in a really nice way of where it's going to be deployed, what they call infrastructure and budget. So you can think of somewhere more clinical with a high budget is going to be more like a hospital or a private pharmacy at the ICU, military-based clinics. They're going to have trained operators and have a budget to actually purchase diagnostic systems. Now, if you move it out to the field, you still have a high budget if you're working with the DoD or maybe an Army base, or you're putting something into an ambulance. It's got to be more field ready, right? You're not necessarily going to have highly trained operators like you would have in a hospital, but you still have a higher budget to work with. Moving down, if you have to be more sensitive towards the budget, but you still have a clinical setting, this might be more nonprofit work or primary care settings. And finally, the last use case is what we would call like low resource settings. So it's out in the field and you have to be very conscious of the budget. There's a focus on global health and self-testing here. And so any kind of system you design, you have to be thinking about where it's going. So are you going to have trained personnel? Is it going to be portable? What kind of handling is it going to overcome? Are you going to have electricity? If you have low resource setting, you might have access to electricity or refrigeration. You got to think about the cost. It's not just how much the system costs, but how much is someone actually going to be willing to pay for it? Is it better than the system that they have in place? Again, will the operators be trained? I don't like this last bullet point I wrote because that's more assay dependent. I think what I meant more is the readout. So you can think about a pregnancy test or an at-home COVID test. That's just a very distinct yes, no kind of screening tool, right? Versus something like RT-qPCR or a clinical pregnancy test where the operator is the one actually seeing the result from this molecular diagnostic test and then telling the patient. And so you have to think about which one that your system is going to be for. And so once you have kind of these answers, you can start to build these microfluidic systems that have your flow drivers, your sample prep, any kind of labels or reagents that you need, your detector, your power source, and you can systematically go through each one and decide what is best for your system for your application. So I'm going to now transition to more of a survey of topics of what's in the literature in terms of these different requirements that I've put out on the side here, starting with pumping or flow driving. Now, really the important considerations to think about are what flow rates you need, what volume do you actually need to pump, what kind of pressure do you need to overcome based on the design of your chip, as what fluid is being pumped, pumping saliva versus pumping PBS versus pumping blood, different reagents. You got to consider what the viscosity and what your either sample or fluid being pumped is. So these common mechanisms like positive displacement pumps, syringe pumps, peristaltic pumps, piezoelectric pumps. For centrifugal, again, you're having more of a spinning motor that's spinning that chip on its center axis. You can have electroosmotic flow where you're putting in electrodes and having the charge on the wall pull those opposite charges in a certain direction, and you need a power supply to do that, or capillary action. Anyone familiar with the COVID tests know about lateral flow assays, allowing capillary action to basically wick that fluid up those paper strands and reach its final destination. And this is a nice graphic of what the different kinds of pumps look like. But I would also like to say there are different ones that can be found commercially. Syringe pumps and flow controller pressure controls, places like Fluigent or L-Flow are great resources to see what's out there for in terms of pressure control systems. You can also find micro pumps. These are great for microfluidics specifically, being able to have the right flow rate and pressure to overcome microfluidic back pressure. However, again, thinking about different applications in the literature, there's actually a lot of work being done on using someone's finger to push fluid throughout a microfluidic. This is great if you don't have electricity, being able to basically push the fluid, create a pressure and push the fluid to where it needs to go. There's also an example here of a micropump being integrated into a very small footprint device. As an example, being able to use one of these kind of micro pump-like things being integrated into a system. So you have your pumps, but what are you actually pumping? You need your reagents for your assay. So what are your reagents? It's going to be based on what assay you're running. If you're thinking about reagent connection and actually housing your reagents on your motherboard or microfluidic device, you got to think about what fabrication technique you're using, where in the process are you going to insert those reagents. What are the conditions in the field? Will you have refrigeration? Will you not? Will it be humid? What are the conditions? What kind of shelf life are you looking to meet? And what kind of pumping method? So two that are basically single use that can be integrated in are things like blister packs or stick packs. Blister pack, you have your reagent basically in a small volume, little blister pack here, kind of like how you would pop out maybe pills from a pill container. You basically have kind of little kind of metal needles that would stick up and basically puncture that burst pack and allow it to flow through the microfluidic device. Stick packs, on the other hand, have a seal that when they reach a certain pressure, like when they're being spun around in a circle, they'll break through that seal. So this is integrated into a lab on a disc. So these are all housing different reagents. There's also a lot of cool work on drying reagents on a motherboard or lyophilizing them on the surface, which is basically freezing them and then rapidly vaporizing them to immobilize them on the surface. Now, this is actually a COC chip, and they lyophilize chemiluminescent substrates. This is actually fairly cool, and then it plugs into a smartphone for detection. And then drying reagents, this was using a porous structure to immobilize, I think it was antibodies or some kind of capture agent on this porous membrane. And so these are kind of different techniques, and you can't use them all necessarily in the same system. It's all going to be based on the fabrication and where you want to deploy your system. So you have your reagent, but how are you actually controlling the fluid throughout the valves? And throughout the system, and that's where valves come in. Generally, microvalves are classified whether they're active or passive. Active, you can think of you're actively opening and closing a valve. Passive is more like under the right conditions, fluid will flow through that area, but it's designed so that only under certain conditions it will. So there's again, the considerations, how many actuations do these valves need to undergo, what the fabrication technique is, what your materials are. Some of these valves require specific materials, what your fluid control method is, what kind of pressure drops do you need to be able to maintain or hold to, and do you need any additional components and what those components are. A great example of a single-use kind of burst valve, it's not really a burst valve, but is a paraffin wax valve. So during the fabrication process, they immobilize these paraffin wax, basically plugs in the microfluidic, and then they heat, when they want to open the valve, they heat it up. The paraffin wax basically kind of melts a little bit and enters this wider region where it basically allows fluid to now openly flow through that valve. Now you can't close that valve. Once it's open, it's open. That's kind of an example of a single-use valve. More of a passive valve would be a capillary valve. These are more common in centrifugal microfluidics. So you basically have a meniscus here, and that going from a thin area to a wide area, the fluid doesn't necessarily want to basically breach this wide region unless you reach a certain pressure or rotational speed. And so this is kind of more of a check valve. So if you increase the speed a little bit, you can basically push past this area and fluid will flow to the other side. Some of the most famous valves are these quake valves. out of the Quake group. This was in Science in 2000. It uses a multi-layer lithography method with PDMS, basically create these cross-hatched layers. So if you can imagine, the horizontal line here would be a fluidic channel, and each of these cross sections here would be basically a pneumatic line, where if you wanted to close this valve, you could basically push down on this line and you would stop fluid from flowing the rest of the way here. And these Quake valves are some of the most famous kind of microsystems valves. But there's a lot of work in other kind of mechanical valves, any kind of mechanical actuation you could think of, piezoelectric, heat. There's shape memory polymers out there that if you heat them up, they'll change shape and open or close valves, electrical valves, magnetic. There's all different kinds out there, and it's all, again, based on your system. One I do want to highlight, because it's one that we've used in the lab, is pneumatic actuation. So what you have is a fluidic layer with your fluidic channels where the fluid actually flows. And on the bottom here, you have a pneumatic layer where you're attaching gas pressure or vacuum. And when you, sandwiched in the middle is this membrane valve. That's kind of the crucial part, is this membrane layer that when you pull vacuum, you're actually pulling that membrane valve into the displacement chamber, allowing fluid to flow through to the other side. And when you apply gas pressure, you're pushing that membrane onto the valve seat and closing the fluid. So this is where we would consider more of a modular approach, because you can have basically everything housed, the gas pressure and the vacuum lines all housed off the microfluidic basically chip, and all your membrane valves are within the motherboard. This is an example, the SMART chip, which we've used for both circulating tumor cell and circulating leukemia capture and analysis. In the past, we've used PDMS. PDMS has a few limitations that are commonly known, absorption of small molecules, hydrophobic recovery, and it just doesn't necessarily scale well. So we introduced cyclic olefin copolymer membrane layer. It's actually an elastomer version of the COC grades that Maggie talked about earlier. And why did we use these two materials? It has to do with their properties, their mechanical properties. So what you see here is Young's modulus, which is basically how flexible a material is. With your more rigid thermoplastics, you can see compared to elastomers. Elastomers are, as their name suggests, elastic. They're very flexible. So what you can see here is the Young's modulus. I should have included a rigid thermoplastic, but they're in the thousands of megapascals versus these elastomers, which have low Young's modulus. They're extremely flexible. Also for valves, you have to consider the elongation at break. They're very high, so they can be extended very far beyond their starting size. And so that gives you a lot of freedom with valves. So there's lots of thermoplastics out there. You have these slides. You can look up these different thermoplastics. A lot of them out there are PDMS, but there's a lot of work out there in the literature to replace PDMS with thermoplastics. Next is interconnects. How do you actually connect these different fluidic components together? Commonly, you can think of O-rings. Those are really hydrodynamic seals. This is an example where you have these interlocking features, and the seals are basically sealing in these two substrates. You can also have press fit connections, basically peg in a hole type ideas, as well as a very primitive well idea. You just are pipetting your reagents in and allowing the sample to be kind of housed here. Now, there's a lot of different connections for modular systems. Like I mentioned earlier, this Lego connection, tubing, O-ring, adhesive connections. But one I'd like to highlight from our group comes out of Louisiana State. This is the gasketless superhydrophobic fluidic interconnects. The idea is you have a hydrophobic coating on the sides of the two microfluidic devices, and you're basically stopping the fluid from breaching out to the sides, and you're creating this liquid column that goes from the one device to another. Just like I said, creating this liquid bridge. And so what you can see from top to bottom and left to right is as the fluid starts to come from this top substrate to the bottom, it's creating this basically column of liquid and not flowing out from the side. Now, if you have a hydrophilic surface, it would just coat that bottom side here, but it's only staying within this region. And this is a video. So you can see where the hydrophobic coating is. And when you drop water, it basically beads up and flows off the surface. So it doesn't require any additional components like gaskets. It kind of standardizes these interconnects. So it can basically be used in any case for these modular components. You have a set gap distance. You're really getting rid of any kind of dead volume. And the one disadvantage you have to just be cautious of and be aware of when you're designing the system is a high pressure can destroy the seal, but knowing that up front, you can design around that. And so what they did was put together this test platform where you basically have a top plate and a bottom plate where the fluid is going to flow from the top to the bottom and back up. So this video you'll see looks fairly normal. You can see where these small kind of round areas are. That's where the top and bottom are connecting. And this looks great, as it should. Fluid is just flowing through as you'd expect. But what I want to show next is without this hydrophobic coating, liquid will just start to leak out the side. So you'll watch right here. As soon as that blue dye starts to come through, ooh, it just goes everywhere. Okay, so that's super hydrophobic coating. Without it, you get leakage between those two. So the next thing, when you're increasing complexity, you're generally going to have some kind of nucleic acid amplification. And what you need for that is generally heat or thermal management. Now, I'm not going to get too much into thermal management, but there is a great review article if you're really interested that goes through all the different components you need for heat management, controller, programmer, driver, what's actually heating, how are you detecting what that heat is, and how are you controlling that heater to remain at that temperature. Now, in general, what you're doing is applying a voltage and forcing electrical energy into thermal energy. And here are some examples. Cartridge heaters, polyamide sheet heaters, thin film heaters are all great examples of heaters that are out there. Again, they do a really nice job of outlining the different heating techniques that are actually being used in microfluidic systems now. And some of the considerations to draw your attention to is what range of temperatures you actually need, how long you need to maintain at that temperature, what kind of response time are you looking for, and also importantly, what material you're using. So I forget who introduced it, but someone said something about making chips for PCR in PC because it had a high TG, so it wouldn't start to deform under these high temperatures you need for PCR at 95 degrees. So there's a few other different examples in the literature that get away from these similar heating techniques. One is an IR laser using an infrared laser to actually heat up the fluid. Another is integrating electrodes into your microfluidic chip and using a PCB board to basically heat those electrodes and thus heat up whatever in your microfluidic chip. Another are copper plates. Having heaters attached to your copper blocks. Copper blocks copper is extremely thermally conductive, and so any kind of heat it gets, it'll transfer. So with copper blocks, I want to bring up again this genosensor where different thermal temperatures had to be reached. This is an IR thermographic image of the different temperature zones from 65 all the way up to 95 degrees. And what's really great is you can do finite element thermal simulations using either ANSYS or COMSOL simulations. So on the bottom here are the copper plates held at these different temperatures. And based on the basically thermal conductivity of your substrate, you can see what the actual heat distribution is. So what we're using for our systems are these copper plates. You have a thermocouple inside, you're attached, and basically what you're doing is attaching this captain, that's that polyamide heater. You attach those to your modules, and then that connects to a PID controller. PID controller is just making sure that it maintains the heat that you're setting those different copper blocks to. So the final one I'm going to go through is power. I'm kind of linking that with voltage and electrodes. So obviously what needs power, everything I just talked about, the heaters, the pumps, the valves, any kind of optics you have, whatever your detector or sensor is and any kind of analysis that you're doing on the system. But again, I like review papers. And so this is another really nice one that kind of links electrochemistry, biosensors, and microfluidics. If you're using electrochemistry, generally you're applying electrode or some kind of voltage to your sample and basically differentiating what's in that sample based on what you're getting out. Amperometry, cyclic voltammetry, impedance spectroscopy are all great examples. And if you're looking to integrate that into microfluidics, you have to think about how you're actually applying those voltages or electrodes into your system. And this is showing that people have been thinking about this for a while. This is an early version of an integrated system that was linking PCR to separations. So this is capillary electrophoresis. So they were amplifying and then had an electrode that was allowing for electrophoresis within their microchannel to do capillary electrophoresis. And so being able to apply voltage, connect all the power, and apply electrodes for different kinds of assays are all very important. Just some cool examples are things like a portable culture counter. This is a fairly recent paper. They basically made, rather than a fluidic motherboard, kind of a fluidic circuit board. So they could connect their reagents and then do electrical readout using their culture counter, kind of like a resistive pulse sensing, all with this very small PCB board. There's also work being done on compact power supplies. So connecting Bluetooth to PCB and then connecting that PCB to a micropump, a piezoelectric micropump to make this really small assembly that can be basically battery powered. And then finally, in order to integrate those electrodes into your microfluidic system, being able to pattern the electrodes, this is a thermoplastic, and they patterned electrodes on it using a titanium layer. And so these are all kind of important considerations, again, when designing your system. So there's lots of review papers out there. I put them all here. But now what I'd like to do, I think I have time. Okay. Is go through a case study for one of our integrated systems that we're currently developing in the center. So stroke is one of the leading causes of death and disability in the US. There's two different kinds, and it's based on whether you have a burst blood vessel or a blocked blood vessel in the brain. Hemorrhagic is a bleed, and ischemic is a blockage. And in either way, you're cutting off oxygen and blood from getting to the rest of the brain. So it's highly pervasive and costs a lot of money to the US healthcare system. So why is that? It requires computed tomography, CT scans, or magnetic resonance imaging techniques. So you have to basically see that either blood is being blocked or burst in the brain. And these are either sensitive to ischemic or hemorrhagic just based on basically the physics of how CT and MRIs work. But why is that a limitation? Now we're in Kansas. If we think about Western Kansas, you don't necessarily have 24/7 access to MRI or CT scans, let alone having the trained operators there 24 hours a day. Stroke doesn't differentiate between 12 a.m. or 12 p.m. So there's low availability. MRI is not necessarily widely used for stroke diagnosis right away. And so one of the sayings in stroke diagnosis is time is brain. So every second that someone goes without oxygen for their brain cells are dying. And so having any kind of time constraint on diagnosing stroke is definitely a negative. And so there are treatments out there. Hemorrhagic is usually surgical intervention. Ischemic is this RTPA, plasminogen activator, basically breaks up the clot and allows blood to flow through. But people have problems accessing it because it has a tight time constraint. So with collaborators at SUNY Downstate Stroke Center, there's been a lot of work from our center on developing an assay that detects actually changes in the blood sample of ischemic and hemorrhagic patients. So again, like Maggie introduced, looking at extracellular vesicles and looking at the mRNA expression changes. So mRNA expression changes are kind of like a fingerprint to what proteins were being produced in response to this traumatic event that happened in the brain. So your body's releasing all kinds of immunocascades in response to the ischemic or hemorrhagic event. And so these mRNAs are basically precursors to those proteins. So now if we're thinking again back to where we would like this to fit in, we're generally thinking of more of a clinical setting, being very conscious of the budget as well, making it very accessible for more rural areas. So if we want to build a system, we went through all these different processes and decided again on a modular approach. It's fairly complex, so we'll need a multi-step assay. We use a minimally invasive sample again to so that people can access the diagnosis, and then point of care setting, again looking more in a clinical setting use for this diagnosis. So I'm not going to go step by step through this whole assay. It's a lot of molecular biology, but basically what we're going from is selecting out of the EVs, getting out the RNA, converting it to DNA, specifically tagging the DNA that's from those mRNA markers that I mentioned earlier, and then using RPS to basically differentiate how many of those mRNA transcripts or cDNA transcripts that we actually have. So in the lab, we have all these different modules that do each of these steps. I've kind of color-coded what each of these modules looks like to their different molecular steps. So these have all been optimized and are running in the lab currently, but the idea was how do we actually build an integrated system from those? So this is courtesy of Daniel Park at LSU. He's done a lot of work to design this platform to connect these 6 different modules. So I'll go through very quickly the different requirements and how we thought about this from a systems engineering standpoint and how we're planning to build this. So again, we chose a modules on motherboard technique. This motherboard will be all disposable, all plastic, where we'll have basically all the peripherals housed in this separate instrument. So all you're doing is plugging in this little microfluidic chip onto your instrument, and it will all be autonomously flowed through without a trained operator there needing to pipette anything. Additionally, what this allows for is a plug and play technique. So what I'm not getting into is this is actually a universal platform. So we can plug and play our different modules that fit into this same motherboard for different assays. And so basically having all the peripherals housed separately, all you're changing out is your different modules and motherboard. So obviously we need to be able to connect all these different modules. So all the fluidic channels are housed in this motherboard. Again, we're going to need heat on these 3 modules. So polycarbonate was chosen as the motherboard material because it has a high TG and can withstand that. We chose this copper block heating. It allows for easy integration for this instrument. You're not changing any of the fabrication of any of your different modules. All you're doing is making an all plastic microfluidic chip, and all your peripherals again are housed outside. The membrane valves are one I didn't actually introduce, but it's a mechanical actuation rather than using gas pressure. You're actually using a physical force to actually push on that membrane layer to close off the valve. And we use these tiny little solenoid valves. And the plunger is actually what's pushing on that elastomer layer to close off that valve. Again, we chose COC because we can mass produce it. We'll need multiple actuations. I should show where those 12 valves are. These need to be repeatedly open and closed, so something like a burst valve wouldn't work. This is a very easy fabrication technique that we've developed. And since this is technically a universal system, these valves can hold to a variety of different pressure drops. And so whatever modules you put in, they'll still be able to hold to those different pressures. One thing I didn't get into is mixing. Like you've heard before, mixing in microfluidics is hard. I've put a great review paper again on the different mixing methods. The one that we're using here is just on the motherboard. It doesn't require a different module. It doesn't require any kind of other, any outside peripherals like some of the other methods do, and it is adequate for our system. So it's all housed on the motherboard. Where is oh, sorry, I'm missing my little yellow box here around my reagent connections. So all the reagents are housed actually off the motherboard. And this was, again, because we have this universal platform, you can easily change out which reagents you want to use. And we're connecting that with micropumps. So these are easily integrated into this basically instrument and can be controlled autonomously. These in particular match the flow rates we need. They allow for that small footprint, And again, we can connect a variety of different reagents based on our application. Now, while we're moving towards the hydrophobic coating, we're using just conical ports for now to connect the modules to the motherboard. Again, my little squares didn't show up, but anywhere you see I in and I out are where these modules basically connect through. This is really easy for fabrication, and it all fits within the same dimensions. So this is what this motherboard actually looks like. You can see the 6 modules and the motherboard highlighted in green here. I just have a short video. You can see it actually moving through. This is using the solenoid valves and for now just a syringe pump, but you can see the fluid start to move throughout where we want it based on where we have our valves open and closed. And the point I want to get to, or right after it leaves this module, you can see it's going to take a right turn rather than a left turn because this valve right here is closed. So we were able to show fluid throughout all 6 modules using this system and our different solenoid valves. So with that, I'm going to conclude. Hopefully I gave you an overview of what systems engineering is, what goes into designing these platforms, what you have to consider when you're thinking about your different applications, and how we're doing it in the system with the COC valves, hydrophobic coating, and different thermal management techniques. I don't know if I went over by a lot, but thank you for your time, and I'll take any questions maybe later.
Yeah.
Yeah, you're spot on. Throughput and flow rate are kind of directly interlinked, right? So if you have high pressure, pushing it at a higher flow rate is going to increase that pressure. So I didn't include what the actual pressure drop of that super hydrophobic coating is. I don't remember it off the top of my head. It's in that paper and you guys all have that reference. But one of the things you could do is put those fluidic interconnects I showed, those kind of peg in a hole conical ports, and those hold to very high pressures and you can run much faster. So it's depending on, I guess, again, you got to think about how much volume you actually want to pump at what flow rate to decide that. I mean, that's, yeah. Yeah, application, where it's being sent, how much fluid do you need to pump to go where, under what pressure. It's all stuff that's got to be done upfront to make those decisions. I went over that very quickly because I was worried about time. We're using the conical ports for now. I believe we're moving towards using the super hydrophobic coating, the gasketless coating, but for now, those interconnects just uses basically this polymer tubing, and it has this conical shape that we've reported on before, that when you press it in, it basically deforms to this conical shape and really locks in and holds really well. Capsule, thank you. Yes. Thank you for the question.
Which brings us to another refreshing presentation by Dr.